So, our group has focused on trying to understand the mechanisms of interaction between the immune system and ovarian cancer. We made the observation several years ago that patients whose tumors are highly infiltrated by T cells, especially CD8 positive T cells, have a better outcome compared with patients with poor infiltration. This prompted us to begin to search for the targets of recognition by the T cells. So what are these T cells recognizing? And this led us to search for antigens recognized by the T cells. We focused our efforts on a family of antigens called cancer testis antigens. These are a unique class of antigens that are usually expressed by tumor cells, not by normal cells, except the adult male testes. The major antigen that we focus our efforts on is an antigen called NY-ESO-1, and this became the prototypic antigen for our cancer vaccine trials. So we developed vaccine approaches whereby we wanted to prolong remission in ovarian cancer patients because, as you know, ovarian cancer typically, most patients typically respond very well to initial therapies, but ultimately the majority will relapse, and once relapse occurs, it's difficult to control. And so we focused initially on patients who are in remission from ovarian cancer. We vaccinate them to generate anti-tumour immune responses in order to potentially prolong the remission time, or in fact, completely prevent relapse of disease. So these were our initial trials focusing on this antigen target, and our results demonstrated significant promise. However, it became clear to us that for when these patients ultimately relapse, the question is, what are we going to do for these patients? So we studied mechanisms by which ovarian cancer escaped from immune attack. Some of those mechanisms include the role of the checkpoints, PD1 and PD-L1. We published on this about 6 years ago in ovarian cancer. Another mechanism is an enzyme called indolemin 2,3-dioxygenase, IDO. These enzymes catabolizes tryptophan, and T cells are very sensitive to tryptophan. When T cells sense tryptophan, that there's no tryptophan in the environment, they are not able to proliferate, and in fact it leads to arrest of their proliferation. So once we identified these pathways, we began to combine with our vaccine strategies in order to overcome any attempt by the tumors to escape from immune attack. So we have ongoing clinical trials where we're not only vaccinating these patients, but also counteracting the escape mechanisms by the tumor by blocking IDO and blocking the checkpoints. One of our major recent efforts has been a focus on adoptive cellular therapies. Adoptive cellular therapy is able to mediate regression of large established tumors, both in animal models as well as in the clinic. But the approach that we have taken for adoptive cellular therapy protocols is really to re-engineer T cells. There are different ways of engineering T cells, as many will know. One group of engineered T cells are called CAR T cells. But we have focused our attention on another method, which is to use T cell receptors for engineering the T cells. So these engineered T cells are produced by doing leukapheresis in patients, modifying ex vivo the T cells, generating large numbers, billions and billions of these cells, and putting back into patients. But the story doesn't end there, because as we started doing these clinical trials, we found that although we can induce large numbers of T cells when they enter the patient's body, the frequency of the T cells begin to decline after a while. In fact, usually by 60 to 90 days, you see very low frequencies. So we raised the hypothesis that, is it possible to use young cells precursor hematopoietic stem cells for these T-cell engineering approaches. And this is what we are currently doing. We've shown very clearly in our preclinical models that the younger the precursors are, and you engineer the cells using our T-cell receptors, you can actually be able to provide long-lived cells that are able to last almost for the entire lifetime of the patient or of the mouse model. So we are harnessing this approach in a clinical trial for ovarian cancer patients, probably within the next 9 to 12 months, whereby we will take hematopoietic stem cells from the patients, we will re-engineer them ex vivo, put them back into patients, and allow these cells to be a continual source and replenish the lymphocyte population in vivo and become a lifelong source of anti-tumour effector cells. So what we find is that in animals where you don't use the hematopoietic stem cells, if you adoptively transfer mature T cells, the T cells don't last for long. Whereas when we adoptively transfer the hematopoietic stem cells that have been engineered to bear cognate receptors, okay, the cognate T cell receptors, not only do they persist for long, as the stem cells mature and differentiate into mature T cells, they now bear those receptors with ability to recognize and destroy tumor targets. So I think in the end, what I think is going to happen in the future is that we're going to have to combine all the different strategies. On the one hand, generate very robust anti-tumor immune responses, either using our vaccine approach or using the adoptive cellular therapy, and at the same time counteract tumor escape mechanisms, the mechanisms by which ovarian tumors escape from immune attack. And we are already beginning to do our clinical trials by actually segregating, by subsetting patients based on a knowledge of the signature of the immune landscape within the tumor microenvironment. So that's number one. Number two, we're also beginning to identify patients based on the mutational pattern. Because of advances in genomic technology, it's now possible to rapidly sequence tumors, understand the mutational landscape. So in my opinion, now we're combining the patient's mutanome, the mutational landscape, with the patient's immunome, which is the immune landscape, and together bring them together in order to deliver powerful treatment strategies for our patients. So we've paid very careful attention to this at Roswell Park Cancer Institute. So we've built the infrastructure to be able to do this. So for genetic engineering, you need a GMP infrastructure for rapid manufacture of your viral constructs. So we've developed this. We have the infrastructure for cell production. We have all of the technologies come together within our Center for Immunotherapy that allows us to rapidly move the concepts from our mouse models to the clinic. Because we can rapidly put all of these things together. Of course, it's a lot of work, especially with regulatory hurdles, but it is now possible. And I envision that as we become even more and more familiar with these approaches, it should get easier and easier with time.